Now, do you see the worksheet? You should. I'll make it a little bigger.
Yeah, we can see it.
Okay. Most of you got most everything right until toward the end. Some of the last questions on the worksheet we'll spend a little time on. The first one, which is the following statement, is true about enzymes, speed up reactions, they're not destroyed, they lower activation energy, those are all true. What's not true is that enzymes are consumed in the reaction they mediate. An enzyme is a protein. I think you all got that. Then some of you had trouble with number 5, with the graph. The answer is letter C. This is the activation energy with enzyme. You can see that it's relatively small, and that's because the enzyme lowers the activation energy. So what is the line labeled B? What is that showing?
Isn't that the energy needed without an enzyme?
Yeah, and you can see that it's quite a lot larger, a larger amount of energy, which indicates how beneficial an enzyme is in terms of getting a reaction to proceed. Okay, so that's the answer to number 5. 6, cellular structures are mitochondria, and we'll talk more about mitochondria later. Energy from respiration is converted into ATP, and it's the breakdown of glucose, which is done by mitochondria to provide energy to make ATP. There was a lot of different answers to the next question. According to the second law of thermodynamics, an organism whose metabolic systems are at equilibrium would be dead. If you remember, to keep things alive, because of the second law, because of the second law, there's this inexorable tendency toward disorder, toward entropy. And that's what happens to living systems if they don't have a constant input of energy. So we must eat on a regular basis. We must breathe all the time because we need oxygen. And that's just to keep the energy flowing into our bodies to keep us alive. So anytime energy stops flowing, we're dead. 2 types of chemical reaction. Those that require energy input are endergonic, and those that release energy are exergonic. Well, the enzyme partners, there are actually 5 of these that are correct. Most of you got 3 of them: cofactors, coenzymes, and prosthetic groups. But also, iron is a partner, an enzyme partner, and so are vitamins. They're subcategories of the others. So to get full credit on that, you need to indicate all 5. And then this last question, number 12, some of you got it completely right. In some cases, no, and in some cases, you just reversed what's actually true. So anabolic pathways require input of energy in order to convert small molecules into large molecules or polymers. Conversely, catabolic pathways release energy because they break down large molecules into smaller ones. Or some people said complex molecules into simple ones, any words that had the same meaning. So any questions about any of the answers? Because this worksheet is relevant. Some parts of it are relevant to the test that you'll be taking on Monday. On that issue, remember that test 3 is on Monday, and it will cover cell division, mitosis, meiosis, Mendelian genetics, and energy and catalysis. So it will not cover photosynthesis, which is a topic for today.
Professor, I had a question.
Uh-huh.
So for that last question, the complex or the catabolic pathways and stuff,
Uh-huh.
I put down the small and large molecules, but I was also confused on the slide, like the next slide after that is the flow chart with the different like chemical reactions.
Uh-huh.
Is that something that we could have possibly answered, or is it just the basic small and large molecules?
Those two slides should have been complementary. In other words, there shouldn't be an inconsistency between the two. Are you suggesting that there was?
No, I think when I was answering the question, I just got confused. I wasn't sure, like, it seems so simple to just put small and large. So I wasn't sure if I was supposed to be putting chemical answers that was on the flow chart slide.
Don't make it more complicated than it really is. In this
Okay.
case, it's that simple, small and large, or simple and complex.
Professor,
Yeah.
I emailed you the worksheet. I haven't been able to submit it on Canvas.
When did you do that? When did you email it?
I emailed you on Canvas that I was emailing it to you from my email, and from my email about 7 o'clock last night.
Oh, Did you use campus email?
So I can't upload anything on Canvas. So I emailed you on Canvas to tell you that I was going to email you from my personal email.
Oh, and you've done that? Because I haven't seen it. It's in my personal email.
Yeah, I sent it to olin.yoder@gccd.edu.
That would be my campus address, and I haven't received it. Well, I'll look for it after class. Let me make a note of that.
Okay, thank you.
And if I don't have it in either of my email accounts, I'll let you know.
Thank you.
Okay. Okay, anything else about that before we go on? Well, here we are back again with our overview of now it's not only where we're going, but where we've been. We've been in energy and enzymes. And the reason I have these 4 topics listed together, energy, enzymes, photosynthesis, and respiration, is that they're all interconnected. And it's really hard to discuss one without discussing the others as well. Now, we've talked about energy, what it is, where it comes from, how it gets used in living forms. Enzymes, their structures, their functions, and importantly, their regulation. Make sure that you understand enzyme regulation. And today we're going to talk more about photosynthesis, focus on photosynthesis. But also we'll talk a little bit about respiration, because the two are opposites. The two processes do opposite things in the cell. And so it's hard to talk about one without talking about the other. And there are certain features that are common to both. So we'll talk mostly about photosynthesis today, a little bit about respiration, but then the next lecture will be mostly about respiration. Photosynthesis. All life on Earth requires a constant input of energy. Remember the second law, says it has to. If energy stops, the organism dies. Source of the energy is the sun, usually. I'll say more about that in a minute. Green plants are the producers. Animals are consumers only. Animals do not produce food. They eat plants or they eat each other. And even carnivores depend on organisms that eat plants. So all the source of energy for life on this planet comes from plants. Food chains always start with plants. There is another process on this planet that can convert, that can produce food. And it's called chemosynthesis, in contrast to photosynthesis. And the difference is that in photosynthesis, here on the left, energy from the sun is the source. So carbon dioxide and water plus sunlight gives us sugar and oxygen. Now, chemosynthesis is the same. Carbon dioxide and water gives us oxygen, plus oxygen, but the source of energy instead of sunlight is hydrogen sulfide. And the products are sugars and sulfuric acid. Now, this doesn't happen in most places on the planet. It's almost entirely in very deep ocean waters that are so deep that sunlight doesn't get there. So these organisms operate entirely in the dark. They have to have another source of energy other than sunlight, and that source happens to be hydrogen sulfide. It's just a chemical reaction. So I don't want to belabor this. Just wanted to make you aware that there is another source of food for life on the planet besides the sun. And these pictures here are of some of the organisms that live in these deep ocean waters. They're very strange indeed. So here's an overview of what happens in photosynthesis. And keep these two things in mind. There are light reactions and there are dark reactions. They both happen in the chloroplast, but in different parts of the chloroplast. So the light reactions involve the green part of the plant containing chlorophyll. Sunlight enters, shines on the pigments, and water is split such that energy is produced and oxygen is released, molecular oxygen. And as a result of this, energy is generated and energetic electrons are generated. And in the same organelle, these energetic molecules go into what is called the Calvin cycle. And here an amazing thing happens. The gas, the carbon from a gas, carbon dioxide, is converted into a solid, a sugar. So this happens in what's called the stroma of the chloroplast. The light reactions occur in what are called the thylakoids. And I'll show you more details of those a little later. So the summary here is that light reactions generate electrons, which are transferred via carrier molecules like NADP to the dark reactions, the Calvin cycle, which generates sugar. And the sugar then, of course, is what we use as food. A chloroplast structure. The top object here represents a cell from a leaf. You can see this internal structures, the nucleus, this large vacuole, and the little green things are chloroplasts. Then you see a blow up of a chloroplast. It has an outer membrane, an inner membrane, a grana, and a stroma. The grana are these stacks of membranous structures called thylakoids. And each thylakoid itself has a membrane and an internal compartment. It's within these grana that the light reactions occur. This is where the pigment chlorophyll is, where light is absorbed. The dark reactions occur in the stroma, and the stroma are the spaces in between the grana. This is where the dark reactions occur, and we have the Calvin cycle, which fixes carbon and makes glucose. So electron transport and chemiosmosis occur in the thylakoids. The Calvin cycle, glucose synthesis occurs in the stroma, the dark reaction part of the chloroplast. Now, is this all making sense so far? I know this is a little detailed and complicated, but it's important that you know it, and you'll probably be questioned about it at some point. Now, I mentioned back here, there are these light reactions and dark reactions. I've just told you details about the light reaction. What about a closer look at the Calvin cycle? Well, here it is. It's a dark reaction of photosynthesis. It is a cycle. The input is a gas, carbon dioxide. Then there's this rather amazing enzyme called Rubisco. It takes carbon dioxide and does a process called fixing, carbon fixation. The definition of this here up in the upper right, process by which plants convert atmospheric carbon from the gas, carbon dioxide, to form solid organic compounds, in particular glucose. So electrons from the light reaction, along with carbon dioxide, are acted on by Rubisco to make a 3-carbon sugar, phosphoglycerate. And to do that, ATP is needed. And that the source of this ATP is the light reactions. Then as the cycle goes on, more electrons, energetic electrons, are needed. And finally, you get to a compound, glyceraldehyde 3-phosphate. That leaves the Calvin cycle and is converted to glucose. So 2 of these 3-carbon molecules can be put together to give a 6-carbon glucose and other organic compounds. So what I want you to notice is the stoichiometry here. The input for the Calvin cycle is 3 molecules of carbon dioxide, which means 3 atoms of carbon. What comes out of the carbon cycle is a 3-carbon compound, and each of these 3 carbons is one of the 3 that went in at the beginning. And I have here the structure of glyceraldehyde 3-phosphate. You can see that it has 3 carbons. Those 3 carbons came from these 3 molecules of carbon dioxide. Okay, I've got here, I thought you might be interested in the structure of rubisco. It's very complex. You can see there are 4 polypeptide chains, very convoluted, intricate, and the active site is here on one side. And this is a close-up of the active site showing the actual amino acids in the structure that bind carbon dioxide. And after binding the carbon dioxide, it gets converted into from a gas to a solid.
I had a question.
Yeah,
So the dark and the light reactions are completely separate. Like so they could pretty much happen like alone. Like
yeah, they can. That's a good question because they are completely separate, but they're also connected and dependent on each other.
okay.
So you can see that the light reactions are dependent on chlorophyll and dependent on intercepting sunlight, photons of light. And this can happen without the dark reactions. The reverse is not true. The dark reactions cannot happen without the light. So the energy generated by the light reactions is what powers the Calvin cycle, provides the energy to fix carbon dioxide and generate sugars, which is pretty remarkable when you think of it, how one enzyme can switch a gas into a solid. Okay, so I said we have to consider photosynthesis and respiration together, at least to some extent. Photosynthesis is really glucose synthesis. It's a metabolic pathway that's anabolic. That means it requires energy and builds up a compound. Respiration is glucose oxidation. It's catabolic. Energy comes out and glucose is broken apart. Okay, redox reactions. Now this is a review. We've gone over this before, these very same words, but I'm going over them again to emphasize the points that are made. Energy is harvested through electrons that are moved from one molecule to the next. These electrons carry energy, some of which will be transferred to intermediate molecules as the electrons travel a downhill journey. The basis for electron transfers is a series of reactions called redox reactions. And we went over this before, and we're going to go over it again. Some substances more strongly attract electrons than others. A substance that loses one or more electrons is said to be oxidized. A substance that gains is said to be reduced. And these two processes always occur in pairs. If something is oxidized, a corresponding substance is reduced. And here's the most simple visualization of that. You have substance A, which is reduced. It has an electron. Substance B, which is oxidized. And if this electron is transferred from A to B, now A is oxidized and B is reduced. And as I said, these things, if one of them happens, the other one also always happens. Now, this is the same thing, but we've substituted, instead of a generic substance, we have an organic molecule that has 2 extra hydrogen atoms. And we have what's called an electron carrier, in this case, NAD, the coenzyme. And if this organic molecule, for example, is glucose, here it is in its reduced state with these 2 hydrogen atoms, which get transferred, and now glucose is oxidized and the electron carrier is reduced, and so it's called NADH plus H plus, a proton. It's a reduced electron carrier. And now I'd like to show you a video to help you have a more complete picture of what happens in biological oxidation. So,
all eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the four complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits two high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal two reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in...
I want to pause there for just a bit and point out that we have these protein complexes embedded in the membrane, and that's where the electron transport chain occurs, going from one carrier to another carrier to another carrier. And they end up with this one on the right here, which was shown earlier as having a rotating part. This is where chemiosmosis occurs. So this is where electrons are transported through this channel from one side of the membrane to the other. And there's an enzyme called ATP synthase, which makes ATP out of ADP. But when we talk about chemiosmosis, think about this, that this complex to the far right here is the the set of proteins that is responsible for chemiosmosis.
In reality, they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
So the video was the intention was to try to help you visualize what a membrane really looks like. And you notice that it was in constant motion. Everything in the video was in motion, the proteins as well as the components of the membrane themselves. So now with that understanding of electron transport and chemiosmosis, ways to produce ATP. There are three: substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. In the case of substrate level, it's a direct enzymatic transfer of a phosphate from a phosphorylated molecule to ADP, and that makes ATP. Now, oxidative phosphorylation is the big one. Electrons from organic compounds like glucose are passed through a series of reactions, the electron transport chain, and you just saw that illustrated in the video, to inorganic molecules like oxygen. Chemiosmosis, which I showed you was the complex at the very end of the electron transport chain, that's what generates ATP. In the case of photophosphorylation, light energy is trapped and converted into chemical energy, again through electron transport and chemiosmosis. And I will illustrate each of these points. Ways to produce ATP, number one, substrate level phosphorylation. You see the reaction here, phosphoenol pyruvate plus ADP goes to pyruvate and ATP. What is pyruvate? It's half a glucose molecule. If glucose is split, glucose being a 6-carbon sugar, if it's split, it will become two pyruvate molecules. So the ratio here is 1:1. 1 phosphoenol pyruvate and 1 ADP yields, along with the enzyme, yields 1 pyruvate molecule and 1 ATP. And as you'll see, this is okay, but it's not nearly as productive as oxidative phosphorylation. Way two to produce ATP, photophosphorylation, produces glucose in the chloroplast. So the pigments of photosynthetic cells trap energy, convert it to chemical energy through an electron transport chain, plus chemiosmosis, plus carbon dioxide fixation. So this represents both light reactions and dark reactions of photosynthesis. So on the left, light enters the cell, is absorbed by chlorophyll, and that splits a water molecule into 2 protons, the 2 H's, and 1/2 an oxygen molecule, 1/2 O2. These electrons that are released by the splitting of water are very high energy, excited, and they can enter an electron transport chain. And now you can visualize all those protein complexes, the electrons passing through them, downhill, losing energy at every step. And at every step, there is a redox reaction occurring. You've seen this before, and you remember that electrons are passed from a reduced state to oxidized. They're passed off from, say, an organic molecule to an electron carrier. And a little bit of energy is released when that happens. And then the electrons are passed on to another molecule. And this goes on and on, so that each step of this staircase is one of these. One of these reactions is each of these steps. And at each one, a little bit of energy is released, so that when you get to the bottom of the stairs, the final electron acceptor is NADP in photosynthesis, and it becomes NADPH. And then the Calvin cycle, the dark reaction, converts this into glucose. And glucose then can be used to power essentially all the reactions in the cell. I've just described that. Way 3 to produce ATP, oxidative phosphorylation. The energy from glucose from respiration in the mitochondrion. So I want to emphasize here and elsewhere that oxidative phosphorylation occurs in both plants and animals. If we go back, photophosphorylation occurs only in plants. Plants are the only ones that have chlorophyll. They're the only ones that can take light energy and make it into chemical energy. But plants also respire. They need to derive energy from glucose, just like animals do. So they make their own food and they eat it as well. So electrons from organic compounds are passed through a series of reactions called an electron transport chain. You can visualize that now. To inorganic molecules like oxygen, that is coupled to chemiosmosis, which generates the ATP as energy is released. And the point about oxidative phosphorylation is that it's very efficient. It makes a lot of ATP from a little bit of sugar. And this picture is to remind you what chemiosmosis is. It's simply the passage of electrons through a membrane, through an area where there is an enzyme called ATP synthase, and that enzyme converts ADP to ATP using the energy from these electrons. So let's look at how oxidative phosphorylation actually works. It's involved with a process known as the Krebs cycle. This is to be compared with the Calvin cycle in photosynthesis. The Krebs cycle starts with glucose, which is split by a particular enzyme into 2 molecules of pyruvic acid, each 3 carbons long. And when that happens, there's a substrate level phosphorylation yielding 2 ATP molecules, starting with 1 molecule of glucose. Also, you'll notice NADH, reduced carrier, is carrying along electrons from this reaction. And as pyruvate goes a step further, it's converted into acetyl-CoA. Carbon dioxide is produced and more electrons. Then acetyl-CoA is the first step of the Krebs cycle. More carbon dioxide is produced, more electrons are produced. These electrons are accumulating on one side of a membrane to a high concentration. Now, the Krebs cycle itself has another substrate level phosphorylation, 2 more ATPs. And that's all the ATP that you get from the dark reaction, except for all these electrons that have been accumulating on one side of the membrane that can now enter into an electron transport chain and end up at chemiosmosis. This is very efficient, and you see 34 ATPs are made starting with 1 molecule of glucose. So the 34 that are made from oxidative phosphorylation plus the 4 substrate level ATPs gives a total of 38 ATP molecules from 1 molecule of glucose. So that's the significance of the Krebs cycle of the that occurs in the mitochondrion. Remember, this occurs in the mitochondrion, not in the chloroplast. But since both plants and animals have mitochondria, they both do this process. Now, this table just compares oxidative phosphorylation with photophosphorylation. In the case of oxidative, it's a process that produces ATP using enzymes and oxygen. It's the last stage of aerobic respiration, requires oxygen. Photophosphorylation is the process of ATP production using sunlight. So oxidative phosphorylation, the source of energy is sugar. In the case of photophosphorylation, it's sunlight. The energy source, in case of oxidative, it's glucose. And as I just said, in the case of photophosphorylation, it's sunlight. The location, oxidative phosphorylation occurs in mitochondria. And remember, mitochondria are in both plants and animals, whereas only plants have photophosphorylation, and it occurs in the chloroplast. Oxidative phosphorylation occurs during cellular respiration, whereas photophosphorylation occurs during photosynthesis. And the final electron acceptor is oxygen in the first case, and an electron carrier, an ADP, in the case of photophosphorylation. So I've summarized here at the bottom the two. Photophosphorylation starts with light and water, and along with electrons and carbon dioxide, ends up with glucose and oxygen. Oxidative phosphorylation starts with those end products, glucose and oxygen, and yields energy and ends up with carbon dioxide and water. So when we respire, we use oxygen to oxidize our food, and we give off carbon dioxide. I'd like to say a bit more about the Krebs cycle. Again, this is part of respiration, and I don't want to emphasize it today, but it is something we have to consider along with photosynthesis. Now, this is a review of what I showed you before. The Krebs cycle starts with glucose being converted to a 3-carbon molecule, pyruvate, which is then converted to another molecule, acetyl-CoA, which is the starting point for the Krebs cycle. The Krebs cycle produces carbon dioxide and a lot of protons, a lot of electrons, which then go into an electron transport chain. There's one substrate level phosphorylation here. As you can see, when it's broken down, these are all the molecules. You don't have to know these. Just know that the Krebs cycle is a whole series of molecules that are converted from one to the other as the cycle goes around. And there's another substrate level phosphorylation at this point. So the function of the Krebs cycle that oxidizes glucose derivatives as well as fatty acids and amino acids to carbon dioxide through a series of enzymatic steps. And the result is high energy electrons from these oxidized fuels are transferred to electron carriers in the electron transport chain. And that's where most of the energy comes from, not from the Krebs cycle itself, from the electron transport chain that gets its electrons from the Krebs cycle. Now, this is an interesting point. Many of these intermediates in the Krebs cycle are building blocks for polymers. Therefore, this cycle is both anabolic and catabolic. On the one hand, it's catabolic because it breaks down glucose and derives electrons from it. It's anabolic in that some of the intermediates are building blocks for other molecules. It's important that you visualize where in the cell these things take place, these various different processes. Now, there are 3 names for this so-called Krebs cycle: the TCA cycle, Krebs cycle, citric acid cycle. They all mean the same thing. In eukaryotes, occurs in mitochondria, in both plants and animals. And this is a drawing of a mitochondrion. The inside of a mitochondrion is a complex of membranes, and there are spaces between the membranes called intermembrane space. The electron transport chain and chemiosmosis occur in the inner mitochondrial membrane. It's highly convoluted, but you saw in the video what this actually looks like. It's highly dynamic, constantly in motion, and it's full of, the membrane is full of protein complexes. Well, this just makes a point about food, catabolic pathways. The things we eat are proteins, polysaccharides, and lipids primarily, and they end up being converted to acetyl-CoA, which is the starting point of the citric acid cycle, which is the generator of much of the ATP that we use. Oh, here they are again, comparing them. Substrate level phosphorylation, so 1-for-1 process. Oxidative phosphorylation, which is very efficient. You get a lot of ATP from a little bit of glucose. And then photophosphorylation, which has many of the same processes as oxidative phosphorylation, but it starts with light. It's powered by light instead of by glucose. Now, this is something that you'll want to know at the level of being able to recognize what part of the process is associated with each of these cycles. So you'll need to study this on your own. The definition of these cycles, the Krebs cycle is part of the aerobic respiration. The Calvin cycle is part of photosynthesis. Where are they located? Krebs cycle in the mitochondria, Calvin cycle in chloroplasts. Krebs cycle is catabolic. It may have anabolic components as well, as I mentioned earlier, but it's primarily a catabolic process, whereas the Calvin cycle is entirely anabolic. ATP and carbon dioxide are produced in the Krebs cycle, whereas carbon dioxide and ATP are used in the Calvin cycle in photosynthesis. The Krebs cycle takes place in all organisms, plants and animals, whereas the Calvin cycle is only in plants because it's part of photosynthesis. Glucose is oxidized in Krebs. It's created in the Calvin. Krebs cycle occurs in the presence of oxygen. In fact, oxygen is required. It's not involved in the Calvin cycle. Now, I'd like to come back and go into a little more depth on the electromagnetic spectrum. How can we capture and make use of energy that is available in the electromagnetic spectrum? Photosynthesis is the process, and remember, in this a wide array of wavelengths from very long to very short, there's this one small window, what we call visible light, because we can see it, and it's this same range of wavelengths that plants can utilize. They can absorb red light, yellow light, and blue light, but not green light, and that's why they reflect green light and they appear to our eyes as though they are green. And here you can see, when you look at the absorbance of chlorophylls, chlorophyll is the main pigment that absorbs sunlight and starts the entire process of photosynthesis. The blue line is chlorophyll A, the red line is chlorophyll B, and you can see that both of them absorb in the red range and in the blue range, and no absorbance at all in the green range. Now, plants produce other pigments besides chlorophyll, like xanthophylls and carotenoids, and also anthocyanins. And if you recall, I made a point of that when we talked about the electromagnetic spectrum earlier, that these other pigments besides chlorophyll can mask the green light that's reflected by chlorophyll. So you can see a tree that may look red, may look orange or purple, and yet it still has chlorophyll and is undergoing photosynthesis. Normally, it's just that the chlorophyll is masked by another pigment. Now, taking a closer look at where in the plant photosynthesis occurs, start with a leaf. You take a particular mesophyll cell from the leaf. The chloroplasts are within the mesophyll cells. And you've seen this before. I show it again just to emphasize the point, comparing or contrasting the light reactions and the dark reactions. The light reactions occur in the thylakoids, dark reactions in the stroma, which is the space between the thylakoids. See the picture again, the overview and an expanded view of the Kelvin cycle. Again, this is a review. Kelvin cycle takes a gas, carbon dioxide, and converts the carbon into a solid, glucose. So into the Kelvin cycle goes carbon dioxide, out comes a sugar, and this is powered by energy from the light reactions. So the Kelvin cycle can proceed only after the light reactions have occurred. These are details that you don't have to worry about memorizing. Just want to make the point that photosynthesis is even more complicated than I've described so far. There are actually two photosystems. That means two systems based on different pigments. Chlorophyll that absorbs at 700 and chlorophyll that absorbs at 680. These are called photosystems 1 and 2. And it starts with a photon being absorbed by chlorophyll 680. And this absorption of a photon excites these electrons to a high level of energy. They can enter an electron transport chain, which generates ATP, generates energy, and the electrons lose energy as they go down this chain. But all is not lost, because these low energy electrons can enter photosystem 1, get another shot of light, another photon, which excites them again, and they can go on to produce more reducing power in the Kelvin cycle. And we don't need to talk about cyclic photophosphorylation. This is just another representation of what goes on in the light reactions. You have these photons of light coming in. You have the membrane. This is a thylakoid membrane. Within it are embedded these large protein complexes, just as you saw in the mitochondrial membrane. Electron transport goes on within the membrane, within these protein complexes. And what comes out is energy to power the Kelvin cycle, which is what makes sugar. Again, the Kelvin cycle, the dark reactions occur in the stroma. That's the space between the thylakoids. And the thylakoid compartment is where between the thylakoid compartment and outside the matrix is where the gradient is built up to drive electron transport. You see it visualized in another way, and I like this. You've got both the photosystems there. You have the electrons from photosystem 2 going downhill, losing energy, but pumping protons into the intrathylakoid space. But then they can enter photosystem 1 and get re-energized to generate more electrons for NADPH, and that goes to the Kelvin cycle, which occurs in the stroma. Stroma is outside the thylakoid. What I like about this is that it has the chemiosmosis part of electron transport. Electron transport chain generates electrons, and they can pass through the membrane to make ATP. This is where we get a large amount of our ATP. Okay, overview of light reactions. Oxygen is produced as a byproduct of splitting water. Remember, the very first step in photosynthesis is light comes in and splits water into electrons and a half a molecule of oxygen. Solar energy is converted to chemical energy, and ATP and NADPH are used to power light independent, in other words, to power the dark reactions of photosynthesis. The dark reactions are known as the Kelvin cycle. That incorporates a gas, CO2, into a solid, sugar. It's called carbon fixation. And glucose, after it is produced, can be polymerized into a very complex molecules like polysaccharides, for example, starch. And that's how energy is stored in a plant. When we eat a plant, we can take advantage of that stored energy. This is just an overview of the Kelvin cycle. I don't need to describe it again. And the Kelvin cycle in more detail. This is the same picture that we started out with at the beginning, showing the light reactions and the dark reactions of photosynthesis. And that's the end of the show. And we are out of time. So before I forget, let me remind you that there will be no Zoom session on Monday. You have all day to take test 3, but don't show up for class because class time is devoted entirely to test taking on Monday.
Professor, when is that extra credit sheet due?
Let me check. Extra credit is due the 27th. That's a Tuesday, week from yesterday, I think. And it's due the 27th because I want to discuss it in class on the 28th, which would be a week from today. Anything else?
Professor, can I talk to you after class?
Yeah.
I was just gonna ask if you've found any other groups for me yet.
You still haven't had any contact with your members?
I was the one that no one else is. They're both not in the class anymore.
Oh, okay. Let me make a note. We have to figure out what to do about that.
Yeah.
Okay, I'll get back to you about that, Justin.
All right. Thank you. Bye.
Okay.
Thank you, Professor, and we'll see you, I guess, next Wednesday.
Next Wednesday, right. You won't see me Monday, but you will see me Wednesday.
All right, have a good night.
Okay, and I'll keep Zoom going so we can talk. Someone wanted to stay.
Thank you.
Okay, bye everybody.